Hello, hello everybody. Welcome to our last chapter audio recording. Oh, it's kind of sad. This is chapter 11 and it's going to be all about the digestive system, which is one of my favorite systems of the body. So I can't wait to share with you all the different compartments, all the different enzymes, all the different processes that occur as food moves through our GI tract and we're able to break it down and absorb all the nutrients that it provides us with. So without further ado, let's go ahead and take a look at an overview of the digestive system. All right, so here we have our beautiful giraffe enjoying a nice little snack, and we're going to utilize our illustration on the right-hand side to take a look at all the different items that the digestive system will provide us. What we see happening is that the digestive system is basically about 4 different processes. It is about ingestion, digestion, absorption, and defecation. Now, ingestion is literally the act of consuming food. Digestion is breaking down the food utilizing mostly hydrolysis, meaning water to pull the macromolecules apart into smaller monomers. Absorption is now when we take these monomers, which are the nutritional values that we're after, and allow them to be absorbed, meaning that they leave the digestive tract and enter the bloodstream so they can be moved around to all the individual cells in the body. And then last but not least, we have defecation. And defecation means that sometimes when you have food, there's often materials that we cannot utilize, so we need to compact it. So think your fecal matter, compact it, and then basically excrete it out of the body because it has no nutritional value for us. Now, on your PowerPoint, you're also going to notice that there might be some other terminology that we can utilize to demonstrate our ingestion, digestion, absorption, and defecation. All of these are correct as well. So for instance, it says right here that instead of using digestion, we can just simply use the term assimilation to talk about the fact that you are acquiring your nutrients, you are breaking them down into their smaller parts, which is the digestion aspect, and your ultimate goal is to get them to be absorbed, meaning once again that they leave the GI tract and they go into the bloodstream so they can travel to the individual cells. Gestation, which is at the bottom, is then going to take a look at the process of compacting your fecal matter and getting it excreted out of the body because it has no nutritional value for us. Now, in order for our digestive system to be completely successful, what we need to focus on is going to be your gastrointestinal or your GI tract, which basically is one long tube that starts with the mouth and ends with the anus. And what we need to concentrate on is what type of cells, what type of enzymes, what type of neurons, what type of secretory cells, absorptive cells, muscle cells, what are all the roles that the different cells will play. And obviously along the way in encountering the cells, we're also going to be encountering different organs such as the stomach, the small and the large intestines, where we're going to have to focus on what each of them contributes to the purpose of our digestive system. Now, before we go into the actual organs and tissues and different cells, I want to go ahead and show you some of the most important enzymes that our digestive system will utilize. When it comes to breaking down food, we see that our body will basically utilize 2 main categories. It can use what we call mechanical breakdown and chemical breakdown. Mechanical breakdown is when you use a physical force to kind of mix and pull apart the food. So this is, for instance, what you're doing every time you're chewing, or when food falls in your stomach and your stomach is churning it and mixing it. That's an example of a mechanical breakdown using a physical force. However, the other option is to do a chemical breakdown, and the chemical breakdown means that we're going to rely on certain enzymes to obviously speed up our digestive system so that we can get access to the nutrients that we're consuming at a faster rate. Now, the way it's going to work is when you take a look at your food options, we see that the 3 most popular categories are going to be your carbohydrates, your proteins, and your lipids. So on the next few slides, what we're going to do is we're going to take a look at which enzyme is specific for which category, where is this enzyme active, and what is the end goal, aka the monomer that we're after, so that it can be absorbed into the bloodstream and brought over to your individual cells. So on your screen right here, what you're looking at are your carbohydrates. Your carbohydrates are basically items like rice, potato, your pastas. Your carbohydrates are basically made out of sugars. So some of you, instead of saying carbohydrates, you might have come across the term oligosaccharide or even polysaccharide. Well, those are just subdivisions of carbohydrates. At its highest category, when you look at a carb, you're looking at items like starch or cellulose or glycogen or chitin. Many of us are probably familiar with consuming starchy foods. Some of us might recognize cellulose because we're primarily seeing that in the plant cell wall. So any type of plant products you consume, you're consuming cellulose. Chitin is best known as being part of the cell wall for fungi, but you can also find it in the exoskeleton of lots of insects. If you've ever stepped on a little ant or a spider and you hear that cracking sound, that's the chitin that's being fallen apart. Some people like to say chitin. Either way, these are all categories of carbohydrates. And when you consume them in your meal, the purpose is to start breaking them down from a carbohydrate into a disaccharide, which is a two sugar bond, and eventually into a monosaccharide. And to be very honest with you, when it comes to the monosaccharide, we have a few options. We have our glucose, our fructose, and our galactose. The one that your body really loves is going to be glucose. Glucose is what we like to call blood sugar. This is the sugar that your body loves to utilize for aerobic respiration, so it can convert its energy into ATP molecules. Now go ahead and take a look at your chart that's on the right-hand side, and starting on top, you can see that they've listed some of the sugars for you, including some carbohydrates as well as some disaccharides. So a disaccharide just basically means two sugars linked together. It's not as complex, obviously, as a poly or an oligosaccharide, so it's not as large. The best example of a disaccharide is probably sucrose. That's table sugar. That is a mix of your glucose and your fructose together. Also, lactose is another example of disaccharides that we might be familiar with. Lactose is the sugar that we associate with a lot of the milk products, for instance, like yogurt, ice cream, or good old-fashioned milk when we're consuming it. Now, the goal of our digestive system is to take all these different sugars and eventually break it down, as I was mentioning to you before, into the monosaccharide, because in the monosaccharide, that is when we're able to do our absorption, meaning that we can dump it into the bloodstream. In order to do that, what we're going to see happening is that we're going to have to follow our GI tract, starting with introducing the food into our mouth, having it travel down the esophagus until it gets to the stomach, and from the stomach, it's going to enter into the small intestines. Now, the reason you don't see the large intestines mentioned here is because the majority of the work for your digestive system is going to get done by the small intestines. By the time you move down to the large intestines, you're basically just worrying about your water balance and packing up any waste products. So we don't really need to worry about the large intestines right now. What we do need to look at is as the food is passing from the mouth to the stomach, to the small intestines, and being broken down, what are the specific enzymes that we're going to use to speed up this process? And when it comes to our carbohydrates, we're going to need to know two enzymes. We're going to need to know our salivary amylase. You can see it right here. I put a little star for you behind it. And we are going to take a look at our pancreatic amylase. Both of these enzymes are going to be specific for carbohydrate breakdown. Both of these enzymes will work best, are optimal, in a neutral environment. And the reason for that, or the reason I want to point that out, is because when you look over to your chart, notice how your stomach has absolutely no carbohydrate digestion occurring. And that is because when you start chewing your carbohydrates and in your saliva you have amylase, that salivary amylase starts working on the different sugars that you're chewing on. And the minute you swallow it, it travels down your esophagus. Your esophagus is still neutral territory, so you're still partially digesting the carbohydrate. This will then come to a stop by the time it hits the stomach. And the reason for that is because your stomach is the most acidic part of your body, usually a pH anywhere from 1 to 3. And when the salivary amylase gets introduced into this acidic environment, it stops working. It becomes denatured. So there is no enzyme there to continue to break down the carbohydrates. It is only when you now move from the stomach into the small intestines, and the small intestines are once again neutral environment, that we see that we can mix our food with pancreatic amylase. It gets its name because it's produced in the pancreas. The pancreatic amylase is then mixed in together with the neutral environment of the duodenum, which is the top part of the small intestines, as well as all the chyme, which is what we're calling the food at this part, as it's moving its way out of the stomach. And by that means that pancreatic amylase can go ahead and finish the digestion part, meaning, as you can see from your chart, it's able to break it down into disaccharides and eventually monosaccharides. And at that point, what we see happening is as the monosaccharides, so the simple sugars, move along the small intestines, there are different types of absorptive cells. Here you can see them listed, enterocytes, that are able to take up the sugar molecules and in essence move them from the GI tract back into the bloodstream. All right, and this is obviously the goal that your body wants because it wants to use the glucose for its other nutritional as well as energy demand. One thing I also want to point out over here is notice what's happening with cellulose. Cellulose is located all the way at the end of your chart. Cellulose is what we find in our plant cell wall. As you can see, the cellulose will enter the mouth. You're chewing it. Nothing's happening to it. You move to the stomach. It's churning with mechanical digestion. Nothing's happening. And it will pass right through the small intestines and it will remain unchanged. So hopefully as you're seeing this, you're going, wait a minute, cellulose is made out of, is what the cell wall of plant cells are made out of, which means that every time I consume my vegetables and my fruits, all my cells have cellulose wrapped around it. So why am I eating fruits and vegetables if I can't get into the nutritional value that's inside the cell because it's covered by the cellulose cell wall? Well, don't worry. It turns out that our large intestines is going to come in and save the day because our large intestines is going to be filled with gut bacteria, and that bacteria will be able to break down the cellulose so that we can extract the nutrients out of our plant-based foods such as our fruits and our vegetables. But unfortunately, without the bacteria, we would have no luck because as you can see, the amylase is not effective in breaking it down into its smaller units. This is, for instance, why when you consume corn, sometimes when you're chewing on corn, if you don't chew it properly or it moves too fast through your GI tract, the corn will go in the same way as it comes out. Another group of foods that we can take a look at that are in the polymer or large formats are going to be our proteins. So for many, our proteins would include any type of meat product that we're consuming, but you can find proteins in many different items, including like soy or any type of nut-based food. Now, the goal of the protein is to be broken down into amino acids. And in order to do that, we are going to once again take a look at two particular enzymes that are going to be very important to our system. So let's go back to our chart and follow the little stars. And we're going to see that the two enzymes are going to be pepsin and trypsin. There are many others, as you can see them listed below the trypsin, but the two that I really want to focus on are pepsin and trypsin. Now, the other thing I want to show you is look how they're active in different environments. Pepsin works best in the stomach, whereas the trypsin is going to come courtesy of the pancreas and be dropped into the small intestines. The reason I point this out is because they're active in completely different environments. Your pepsin will be available or most active in an acidic environment. So as you start chewing your food, the saliva within the mouth does not have any enzymes related to the proteins. So the proteins will just do mechanical digestion. It will pass through the esophagus, it will enter the stomach, and when it gets into the stomach, that is then when it meets its first chemical digestion, its first enzyme in the form of pepsin. Pepsin starts breaking down the proteins into polypeptides, and as the stomach releases its chyme and it goes into the small intestines, as that chyme gets neutralized, what we see happening is that the pepsin becomes deactivated, and instead trypsin will now take over the role of continuing to break down the proteins into the amino acids. Now, as I mentioned to you before, the trypsin is going to come courtesy of the pancreas and is introduced into the duodenum, which is the top part of the small intestines, which focuses best on a neutral environment. And what we see happening is very similar to our carbohydrates. Once we get down to the monomer, the amino acids, we are going to have different types of epithelium cells within the small intestines that are able to go ahead and take these amino acids and dump them into our cardiovascular system, our bloodstream, so it can be circulated throughout the body for nutritional value. Our third category are going to be our fats, our lipids. Now, for the lipids, we're going to have a little bit of an issue, and part of that is because they are hydrophobic, meaning that they have a tendency not to want to interact with the hydrolysis component of our digestive system. So what we're going to see here is that in addition to an enzyme, we are also going to have to use an emulsifying agent. An emulsifying agent is going to force the lipid and the water components to interact with each other. And our emulsifying agent is going to be bile. Bile is produced by the liver and it is stored in the gallbladder. Both the liver and the gallbladder are able to release bile as the lipids pass through our GI tract. And what we see happening is that when we mix the bile along with lipase, which is the enzyme of choice for our lipids, we can maximize the breakdown of lipids into smaller molecules or their monomers, such as fatty acids and glycerol. As you take a look at your illustration on your right-hand side, you're going to notice it's a little bit different of a setup. And the reason for that is that I wanted to definitely point out the fact that you will have the interplay of the emulsifying agent as well as the lipase enzyme. And in case you're wondering where does the lipase come from, we have two options. We have salivary lipase, which you find in your saliva, and we have pancreatic lipase, which we get courtesy of our pancreas. Now, an interesting part about the salivary amylase is that even though it's present in the saliva and it starts mixing with the food as you're chewing, and it follows the food as it travels down into the esophagus, that salivary lipase actually does not become active until it reaches the acidic environment of the stomach. So it is secreted in the mouth, but it doesn't become active until it reaches the stomach. And then as the food moves from the stomach, the salivary amylase deactivates because it then enters the neutral environment of the small intestines, where it's once again mixed in by pancreatic lipase. And very similar to what we saw before, the molecules will start breaking down into smaller pieces. However, when it comes to lipids, it turns out that their final destination will differ depending on what we are breaking them down into. So on your PowerPoint and on the little box that I just made, it shows you that if you're able to break down the lipids into fatty acids and glycerol, these are able to just be dumped directly into our bloodstream. Whereas the triglycerides, what we'll do with those is that we're actually going to wrap them around little vesicles, courtesy of the Golgi apparatus. These are going to be called chylomicrons, and these will travel in our lymphatic system. And that is then how we will be able to extract the nutritional value out of those. So two methods of transport in this case, the addition of the bile as our emulsifying agent, and then our salivary amylase is only active in the acidic environment of the stomach, and the pancreatic lipase will be the neutral environment, and it will be active in the small intestines. Now, it won't be a comparative animal physiology class without doing some comparison, right? So what I want to do is I want to start introducing you to some simple setups when it comes to the digestive system. And then we're also going to take a look at some different structures and compartments. And then obviously along the way, we'll get into the more detailed, more complex setup of different animal models. So let's go ahead and start off at a very simple level. Let's take a look at our sponges. Now, it shouldn't surprise you that they don't have a very complex digestive system because, as we've discussed, the sponges in the respiratory system and the cardiovascular system, they like to keep everything nice and simple, and they like to utilize a bulk flow of water to really get an extraction of all their different gases and nutrients. So the picture that you see on this PowerPoint should look very familiar to you because you have seen it in your previous chapters before. And what we see happening here is that once again, we see that with our sponges, we're going to do our bulk flow where the water can go ahead and enter within the cavity of the sponge, where it's then encountered by our little choanocytes, which are the flagellated cells. And in this case, what it's showing you is that if there's any type of nutrition or nutritional organisms within the water that's introduced into the cavity, we see that our little choanocytes can go ahead and do endocytosis, allowing the nutritive products to enter into the cell, making these little vesicles. And as they're being introduced into the cell, we're going to start breaking them down. So that's our intracellular digestion that's occurring. Along the way, by the choanocytes, we also see that there are some amoebocytes on the way that can help with distributing of the nutrients throughout the entirety of the sponge. The cnidarocytes have a very similar setup. Once again, nothing fancy, not a very specialized type of digestive system. We once again see that they can use their muscular contraction to introduce water with any type of food particles into their gastrovascular cavity. And as the water is being circulated, we know that they're being exposed to the different cells that are on the side or on the lumen. Now, one thing that I want to point out to you over here is that when you look at your illustration, you're going to notice that they went ahead and they labeled for you the nutritive cells and the enzymatic cells. And the reason this is important to point out is because it turns out that your cnidaria, so your little hydra right there, can do both extracellular and intracellular digestion. Extracellular means outside of the cell, the items are broken down into smaller pieces. Intracellular means just like the sponges, everything is broken down and absorbed inside the cell. And the reason this is possible is because your enzymatic cells will be able to release enzymes into the cavity, and those enzymes can start breaking apart the molecules into smaller pieces. That's the extracellular aspect. And then what we see happening is that the nutritive cells are able to absorb particles as well, create little vacuoles for them, and the particles can then be further broken down by the nutritive cell, and that would then be the intracellular aspect of food breakdown. There is a mesoglial layer that's towards the end of the little gastroderma section. That is going to be filled with a little gelatinous layer, a little bit of mucus, and all that's going to help is going to help with the absorption of the nutrients from one side to the other side of cells when it comes to looking at our cnidaria feeding mechanism. And of course, anything that is unwanted, anything that it can't break down, will go ahead and be exited out of the gastrovascular cavity once the animal is done with it. Now, oftentimes, to assist with our mechanical breakdown of the food, so to create that churning motion and tearing motion to break the food apart in a mechanical sense, we can go ahead and we can look at different feeding structures. So for instance, we can take a look at our mouth, we can look at the use of our tongue to kind of slush the food side to side. Our teeth are definitely good examples of items that we can use for mechanical breakdown. And we can also see that for a lot of animals, usually within the mouth area, there will be a very specific or structure or piece that will contribute to obtaining and processing foods. So I've just listed a few of them for you on this slide. For instance, when we take a look at like liver flukes, we see that they have siphons that they can use to attach to the food and start breaking it down. A lot of parasitic organs will have specialized anchoring organs that will allow them to attach and not fall off as they're feeding off their prey. There are different forms of tongues, like snails tend to have a radula, which is more like a rasping with sharp edges looking tongue. There can be like these longer tube-like tongues like proboscis. So for instance, I put a little picture for you here. This is a moth that Darwin wrote about when he was visiting Madagascar, and he came across these orchids that had this long nectar tube, and he said that there had to be an animal that had some type of attachment organ that would actually fit down this nectar tube. And although he never saw the moth, it was discovered a few decades after his after his death, excuse me. What we see happening is that the moth can actually curl up its tongue, which is about 8 inches long, and when it gets over to the orchid, it will uncoil it and then insert it perfectly into the nectar tube. So this is not, if that's not a prime example of a specialized mouth part, I don't know what can be. We can also take a look at different beaks or jaws. If you look at the picture at the bottom of our snake, the reason that snakes are able to ingest items that are so much larger than they is because their top and bottom jaw can physically separate to increase their oral cavity so that they can swallow these large entities. Like for instance, it's trying to swallow an egg right there, and it will be successful at as well. Another mouth part that's been extensively studied are going to be bird beaks. And this started all the way back to, if we take a look at the stories of Darwin visiting the Galapagos Islands and seeing the different finches and talking about adaptive adaptations and descendant modifications is what he called it. Basically that the beaks that correlated best to the food source would have a higher amount of natural selection and favoritism to the survival of the particular birds. Now, bird beaks usually have lots and lots of keratin. So these are like little epidermal scales. As you can see from the picture below, that is a subgroup of different Argentine birds. And what we see happening is that they all come from a common ancestor, but they have just metamorphosized and evolved completely different, depending on what type of evolutionary adaptations were beneficial to their overall environment that they were located in. In addition to food, we know that the beaks will also play different roles like courtship, vocalization, defense, and grooming. So I just wanted to go ahead and highlight the beaks because many of you probably were thinking about Darwin and his finches and how he was one of the first to kind of point out the fact that we can trace that down. If you can get more food, you can get more nutrients, which makes you more favorable, obviously, to mate with. If we go ahead and take a look at our teeth, what we see is that we have 3 parts to our teeth, and we're basically looking at the crown area, the neck, and the root. So you can see them highlighted right here. So these are the physical items of the teeth. Our teeth are our main way that we can do our mechanical breakdown because it's going to help us tear apart the food and flatten it so it can increase more surface area, making it easier for the digestive enzymes to mix in with. Within the tooth itself, there are 3 layers that I've highlighted for you as well. You have the enamel, which sits on top. This is actually a combination of calcium phosphate crystals in the extracellular matrix. It is considered a dead layer or a dead tissue. And the other 2 layers that are underneath it, the dentin, which is more like a porous support, and the pulp, where you're actually going to have lots of blood vessels and nerves run through it. Those 2 layers are considered a living entity. And as you can see, it's being encased within the bone. So that would be the bottom and the top jaw that is housing it. If we take a look at mammalian teeth, then usually what we see happening is that overall there will be 4 different types of teeth that you'll encounter, starting from the front of the mouth, working our way towards the back. We would see that we would have our incisors and our canines. These are fantastic at tearing up food because of the sharpened edges that they have. And then as you move your way towards the back of the jaw, you're going to notice that the teeth will type widen and flatten out a little bit, and those will be our premolars and molars. These are good for grinding. So usually what we see happening is that the incisors and canines work well when we chew in an up and down pattern, allowing us to kind of like pull apart the food. Whereas if we grind in a side to side pattern, we allow the larger surface area of the molars to truly interact with the food that we have in our mouth, getting ready to grind it. Obviously, there's lots of variations in the number, the size, and also if you have your permanent versus adult teeth that come into play. So lots of variation, but our goal ultimately is to remind you that the teeth is an example of how we can use our mechanical digestion to pull apart the food as we initially introduce it into our GI tract. Now, when it comes to your actual digestive system, what we're going to see happening is that obviously there's an evolutionary history behind it. And usually we see a nice little correspondence to the more complex the organism is, the more complex its digestive system will be as well. And at its most simple basis, what we see happening is that, for instance, in ancient invertebrates, but also even in cnidarians, jellyfish, and corals, we see that the digestive system at its core started with a two-way gut, okay? So a two-way gut basically meant that the organism had some way of isolating its food and its breakdown from the rest of its environment, and oftentimes it could be something as simple as just having like a little internal sac with a lumen, an opening on the middle, and it would have an entry point. Now, with a two-way gut, what we see happening is that the entry point, and you can see it on the illustrations posted, it's labeled as the mouth. And the reason we're going to call this a two-way gut is because the mouth will serve both as your entry as well as your exit point for any of your undigestible type of food. The sac itself could be very simplistic, but also what we see happening is it can also be labeled as a little bit more complex because of the different infoldings that you might see right over here. These little diverticula are a great way to increase the surface area, making it easier for the cells to have access to the nutrients as they're being broken down to. And as I said to you before, this is just a very simple setup of the digestive system with the two-way gut. One way for both entry and exit, that's why it's called a two-way. And as you can see, the gut is just a simple sac. Maybe we can have some foldings to increase the surface area, but very simplistic. But the nice thing about it is that you're still separating your food particles from the rest of the body. In more complex organisms, we're going to take a look at our one-way gut, and that is where your GI system is a perfect example of. So in a one-way gut, what you see is that you have an entrance and you have an exit. And in our case, what we see happening is that the entrance is going to be by the mouth and our exit is going to be by the anus. And as the food is being passed down from beginning to end, it's going to come and encounter specialized regions. Each of them will have their own individual function. Each of them can have their own individual environment, and they will all have their individual purpose that they're serving along the way. Now, the mechanical and the chemical digestion that we've talked about, those will all get incorporated with the different areas that we encounter when we do our one-way gut analysis. And keep in mind that we are going to utilize our different enzymes that we saw at the beginning, so like our amylase, our trypsin, our pepsin, our lipase, and these are all going to speed up our chemical digestion so that we can go ahead and access the nutrients in the food at a faster rate. Can we do our digestive system without chemical breakdown? Yes, you can do it, but it's going to take a much longer time for your body to successfully break them down into monomers. So you're going to have to wait a longer time before you can get access to the nutrients that each of those food particles pertain to. Now, really quickly, what I want to do is, and obviously we're going to talk more in detail as we kind of progress through the chapter, but I want to make sure that everybody at this point is comfortable with the pathway that food will follow as it starts from the mouth and it moves its way over to the anus. So go ahead and take a look at your illustration of the horse, and it's going to be very similar for us. As food enters into your oral cavity, we've already talked about the fact that you have your teeth that will start grinding, your tongue is kind of pushing it side to side. This is all an example of mechanical breakdown. And in your saliva, as you're wetting the food, this is then the start of your chemical digestion because you're introducing it to the different enzymes. The food is then swallowed and will travel through the esophagus. Now, something cool about the esophagus, it is the only organ that you have in your body that passes through both the thoracic cavity as well as the abdominal cavity. Your esophagus will go through your little diaphragm, which is that muscle that separates those two cavities, and it will enter into your stomach. Before it enters into the stomach, it does have to pass through the esophageal sphincter. A sphincter, think of it kind of like a little sliding door. It's going to allow you to open and close the entrance. So the esophageal sphincter will regulate the environment that's in the esophagus versus that of the stomach. Why? Well, because your esophagus is neutral environment and your stomach is your most acidic part of the body. So it's two different environments. And in fact, if the sphincter doesn't properly function, or if you eat anything that might aggravate the sphincter to open up, and the food, the acidity from the stomach moves up the esophagus, that is then when we get good old-fashioned heartburn. So we're going to try to keep those two environments separated at all costs, so your esophageal sphincter will play an important role in that. As food enters into the stomach, then now what we're going to do is, instead of just calling it food, we're going to call it bolus. B-O-L-U-S. I can't spell it. You think I'd get better, right? So bolus is basically chewed up food that has moved its way down the esophagus and is now sitting in the stomach. So bolus enters the stomach, and the stomach is going to grind. It's going to do its chemical and mechanical breakdown because you're going to introduce it to the different enzymes that are sitting in the acidic juice. And whenever the food or the bolus is ready to go, then now what we see happening is that it's going to have to pass through the gastric sphincter. This is also called the pyloric sphincter. And this sphincter is important because what we see happening is, at this point, the bolus has turned into chyme, which is more liquefied, and the chyme is very acidic. So the gastric sphincter is going to regulate that the chyme gets released in small amounts of doses, so that we have plenty of time to neutralize it, because our small intestines is all about neutral pH. And as the chyme is being released little by little, it will encounter the three parts of the small intestines: the duodenum, the jejunum, and the ileum. And the duodenum, its main purpose is to neutralize the chyme. So we're going to see it's going to have lots of Brunner glands that are going to help us with mucus secretion. We're also going to see that this is where the pancreas has its connection, so it can drop off the digestive enzymes in the neutral environment. And as we move from the duodenum, we get into the jejunum. The jejunum is the one that really does all the work. That is where the most digestion and absorption is going to occur within your GI tract. So that is where you get to your monomers, and the monomers are able to leave the GI tract and go into the bloodstream. And then in the ileum, the end part, that is where we start connecting it into our large intestines. And usually that's indicated by a little pouch area right over here, a little cecum. And the cecum indicates the start of the large intestine. You usually have like a little sphincter there as well, the ileocecal sphincter, which is not labeled on this illustration. But either way, as it travels past the cecum, our little pouch, into the colon will get different names depending on which organism we're looking at. But the whole purpose of passing through the colon and any other part of the large intestines is now to regulate your water balance, and also to start compacting any type of unused material. And what we see happening is that will eventually form fecal matter, which will then sit in the rectum right over here until it's able to slide out of the anus through the inner and external anal sphincter. And with that, the story of the food comes to an end because whatever you could utilize is now floating around in your bloodstream so that you can get the nutrition out of it. And whatever you couldn't utilize is passed out through the anus and is disposed of. So that is the story of our one-way gut. It starts in the mouth, down the esophagus, into the stomach, and then into the three parts of the small intestines, duodenum, jejunum, ileum. Don't jump in. That's the saying that they remember the order. Duodenum, jejunum, ileum. Don't jump in. And from the small intestines, it goes into our large intestines, where everything is compacted that we don't need. And then out it goes towards the anus. And the story is done. Now, don't worry, as we go through our chapter, you will see each of these organs again, and we'll talk a little bit more detail. But I definitely wanted to make sure everybody had a nice little overview as to what was happening as food becomes bolus, becomes chyme. Now, in the GI tract itself, what we see is that if we take a look at the lumen, oftentimes there'll be lots of curvations and foldings, and this is all in a way to increase the surface area, the amount of region that the enzymes have to interact with the food as they're being passed through our GI tract. The larger the surface area is, the faster you can get through the hydrolysis of the food. So we could see our body can do this many different ways to optimize our nutrient intake. It can increase the length of the actual gut. On average, our GI tract is over 10 feet long. That is taller than many of us are. And what we see happening is that a lot of that will come from the small and large intestines. And your body can do this because what's going to happen is it's going to coil it all up and keep it coiled by using things like mesentery to keep everything in place within the abdominal region. We can also see that within the lumen itself, we can go ahead and create circular folds, little villi, little extensions, and within the individual cells, you can also have microvilli. So any of these like hair-like extensions that you see pictured on the illustration, all of this is meant to increase the surface area that we have to look at. And a lot of times with the cooperation of the gut length, the circular folds, the villi, and the microvilli, you can basically increase the surface area by hundreds of times more than if you were using a smooth surface. And as I also mentioned, part of what makes our one-way gut so specialized is the fact that we do have these different environments or different compartments that we can keep apart. And some of this, if you're looking at the text, some of you might be familiar with this already because we chit-chatted about it when we were looking at our little horse picture. We're going to use different muscular valve sphincters to allow the food to go from one compartment to the next. So it's very similar to when we talked about the cardiovascular system and the use of valves, is that these sphincters will try to regulate one-way flow at all times. It will also be very important in keeping the food within the individual compartment, especially when you take a look at things like your stomach, the fact that its pH is so acidic compared to the rest of the body. And if that pH were to spill over into your esophagus or into the small intestines, it would literally disrupt those particular environments, reducing the effectiveness, obviously, of your digestive system. Let's see what else we got going on. Oh, I do want to show you a picture where we compare different guts from different animals, so you can see how these specialized compartments will play a role in it, obviously depending on what their ultimate food source is. So let's go ahead and click over to our next slide. Here you go. So here you can see different vertebra gut morphology. You can see our mammals right here with the pig. Notice the coiling of the small and the large intestines. Remember, that's all in an effort to increase the gut length so that we can increase our surface area. Sharks have a spiral valve that they tend to utilize within their ileum part of their small intestines. That's also part of increasing their surface area. With chickens, for instance, you can have a little glandular part of the stomach that's right here in the proventriculus. It also has a little crop. The crop is, how can I describe it? Almost like an outer pouch of the esophagus. It tends to be used for storage of just partially digested food. Usually what we see happening is that the length of the gut will often differ depending on what type of food that animal is used to consuming. In essence, what we see happening is that carnivores tend to have a shorter GI tract than herbivores. So if we see that the animal consumes lots of meat products versus an animal that consumes only plant products. And it seems that the correlation with the longer gut when you're consuming plant products or herbivores is due to the fact that plants have that cellulose layer in their cell wall that does require additional time to be fully broken down so that the nutrients can be extracted from it. Oh, and then also we can take a look at ruminants. So this is, for instance, when we're going to take a look at the digestive patterns of, in this case, like as you can see, the little cow or the steer, goats, sheep, deers, giraffes. What we see happening is that these animals tend to ruminate on lots of plant products. Excuse me, they like to graze. That's the word I'm looking for. On lots of plant products. So their system, their digestive system has been modified to accompany their food of choice. And the way we see happening here is that in addition to having their specialized structure, we see that their stomach has been modified into 4 different compartments. So they have a 4-chamber stomach. And if you look at your illustration, I've highlighted them for you. They are going to be the rumen, the reticulum, the omasum, and the abasum. So these are the 4 parts of the stomach that you would come across within the ruminants. And the way it's going to work is that all the food is basically processed twice through these chambers. So food will go ahead and enter into the mouth. It will pass down the esophagus. And what we see happening is initially the food will make its way to the first 2 chambers, the rumen and the reticulum. So this will be our first stop, the rumen and the reticulum. And in there tends to be lots of fermentative bacteria. So the bacteria is going to help you start breaking apart the cell wall that you have in like the grass products that were consumed. After a while, what we see is that now that it's partially digested, the food will actually go right back up the esophagus, into the mouth. The animal will chew it a little bit more, yum, 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 and then it will once again swallow it, and it goes into the esophagus, and now it goes over to our second stop, which is going to be the chambers that are more towards the back. So that's the omasum and the abomasum. And why do we go over here? Well, this is where we're going to go ahead and find our digestive enzymes. So the digestive enzymes will kick into gear, and they'll they'll start their chemical digestion and finish breaking down the polymers, or continue breaking down the polymers into their monomers. And as you can see, it will pick up the same route as with the rest of the mammals, that it will encounter the pyloric sphincter as it leaves the acidity of the stomach and makes its way over through the small intestines, followed by the large intestines, and anything we can't use, it goes out through the anus. So just to reiterate, the first 2, the rumen and the reticulum, these are going to be the stop where you have your bacteria. And then the 2 back chambers, the omasum and abomasum, those are going to be utilized for digestive enzymes. And in between, the food does go back up the esophagus and is introduced into the oral cavity, where they will chew it again and then swallow it. All right, everybody, this is the end of the first part of the digestive system. Hopefully we can pick it up really quickly and take a look at the second part.